Okay, we've moved on to Module 6, which is Tools of Discovery in Older Brain Structures. So I'm going to start with the old brain, which is common to humans and their primate ancestors, which includes the fight or flight regions of the brain, the limbic system, and the brainstem. And then in the next module, I'm going to talk about the cortex, the outer covering of the brain, which separates humans from other primates. So how do we learn about the brain? Well, we learn about the brain through brain scans, obviously. We also learn through experimentation on animals, including lesioning or putting electrodes in animals' brain and stimulating different parts of the brain and see what kind of behavior is elicited. So we'll talk about imaging a bit, about Fmri research, which is cutting-edge research within psychology. And then I'll move on to talk about the primitive, life-sustaining inner parts of the brain, which include the brainstem and the limbic system, and how damage to the brainstem is a really, really bad thing to have. So, the brain is wonderful. It's a 3-pound pile of mush that sits in the center of your skull. I wish I had a real brain to show you, but I don't. But trust me, the brain is a squishy, mushy pile of neurons, if you want to think of it that way. So how do we learn about the brain? I'll talk about imaging studies. I'll also talk about the inner parts of the brain, the brainstem and the limbic system, which are vital components. And then the cortex, which is mostly in the next module. And a little bit about left, right, and split brains, but they're not too interesting, to tell you the truth. And then some questions about the brain. Do you think that the brain is a sum of its parts, or is the brain actually the way they're connected? There's crossover of function in the brain that I'll mention. What do you think might happen if a particular part of the brain is stimulated? That's very interesting. So people will actually attach electrodes to the brain, stimulate different areas, and see the response. And what happens if a particular area of the brain is damaged? So, investigating the brain and the mind. So how do we move beyond phrenology? Phrenology, if you can recall, was examining the bumps on a person's skull and determining that those bumps correlate to certain attributes within the person. So we've moved way beyond phrenology. Phrenology was a pseudoscience. We're not interested in that. So we get under the bumps on the skull. So how do we do that? Well, we could look at people with brain damage. But that's difficult because you generally don't have brain damage to one particular area of the brain. Brain damage tends to be fairly well distributed. Like a car accident doesn't cause damage to one slight tiny area of the brain. It causes a diffuse axonal injury, which is basically a sheathing of the axons and the neurons that make up the brain. And so that's widespread damage to the brain. So you can't really tell about different areas of the brain controlling different behaviors or localization of function from accidents. Unfortunately, you have to use animals for some of that stuff, or you can use brain imaging in humans, which is one of the ways that I used to do this when I was working in a cognitive neuroscience lab in graduate school. You look at images of the brain, whether that be a CAT scan, which tells you about structural damage. MRI, the same thing. fMRI will give you a very good three-dimensional image of the brain and the activity occurring in different regions of the brain. And then finally, PET scans, which are very interesting to study, or very necessary to study the metabolic activity of tumors. So if the brain isn't working right, you can rely on case studies, like Phineas Gage. Phineas Gage was a guy that worked on the railroad, and one day there was an accident where a metal rail or pipe went directly upwards through his jaw and through the frontal lobe of his skull. So it basically crashed through his eye, through the frontal lobe of his skull, and miraculously he survived, but his frontal lobe didn't work properly. And they noticed a lot of differences in Phineas Gage when his frontal lobe didn't work properly. And that was a really breakthrough in terms of brain research at the time. But we don't have many Phineas Gages walking out there. There's not that many of these people that have selective injury to only one part of the brain caused by accidents. I mean, most people in car accidents, as I mentioned, are either dead or they have diffuse brain injury. So the case of Phineas Gage was interesting and useful, but it's not realistic to expect to learn about the brain by individual injuries such as this. So, split brain patients, sometimes what we do is we sever the corpus callosum, or the band of fibers that connect the left and right hemisphere together. And this is done to stop epileptic seizures. If you have severe epilepsy and the disturbed electrical activity will cross from one hemisphere to the other through the corpus callosum. This tells us some information, but not much. I mean, many split brain patients where the corpus callosum is severed act just like normal controls, normal patients who never had a corpus callosum severed. So we really don't learn that much through these kind of case studies. Lesioning the brain. Unfortunately, this is a way to learn about the brain, and I say unfortunately because it's difficult to do this kind of research on animals. And I've seen a lot of mice and rats with giant electrodes stuck in their head, and lesions of multiple areas of the brain, and even basically killing the animal to examine their brain after a particular experiment. And we do learn things through animal studies, and we do have to kill animals. And as I mentioned back in module 2 or so, the ethics of animal research, it does help us, but it's very difficult to do. And personally, I never felt comfortable working in these conditions where I'd actually execute, or what they call sacrifice the animal, kill them, and then open up their brains and look at their brains. But it's done in many different labs, and it yields many important results, and we can't deny this. So there's I have mixed feelings about animal research, but it does contribute to our base of knowledge, and it does lead to advancements in science and solutions for diseases in human beings. And finally, we can also use certain tools to chemically or numb the brain or magnetically deactivate parts of the brain. This is called transcranial magnetic stimulation. And it's a device, you hold it to the brain, and it interferes with the electrochemical and magnetic properties of the brain. And it basically deactivates part of the brain for a short period of time, for a couple of seconds. And so you can hold it up to the person's brain, deactivate the brain for that region for a couple of seconds, and then see what happens. And this is thought to be generally safe, but it hasn't been around that long to know that it's perfectly safe. I wouldn't probably want to have it done on me many, many times, especially within a short period of time. But so far, it seems safe, and it yields some interesting... So it's kind of like creating a magnetic lesion. It yields some interesting results. However, the machine is very, very expensive, and not many people have this. So it's a lot cheaper to do research on animals than buy all these complex machines, or even do MRIs on humans. Here's Phineas Gage. Obviously, you see the pole that I was talking about basically shot upwards directly through his frontal lobe of his skull, through his eye, and miraculously he survived. But what they found out about Phineas Gage was he was usually a calm, very genial, kind, very nice fellow. Got along well with his co-workers before the injury. After the damage to his frontal lobe, he became very irritable. He became difficult to work with, and not so much a nice person. And the reason behind this is clear, that when you have frontal lobe damage, you stop your inhibitions. So the things that you really want to say actually come out of your mouth. You lose that ability to filter your thoughts and your actions. So, how you want to tell your boss, I hate you, and some other choice words, but you don't do it. Or you may want to punch your boss, but you don't do it. Because, if you punch your boss, you curse at your boss, you probably won't have a job. Your frontal lobe says, hey, stop, don't do this. You're thinking about it, but this is not a good action. When you have frontal lobe damage, you just start acting on that impulse. You become extremely impulsive. And that's why teenagers tend to be more impulsive and pretty bad decision makers, because their frontal lobes are not developed to their mid-20s. And so teenagers tend to act irrationally, and they engage in hasty decisions or hasty judgments and do things that, you shake your head at and you wonder, why the hell did I do this? I mean, personally, I go back to my childhood and think about it and say, why the hell did I do this? This is stupid. Was I crazy? No, I wasn't crazy. I had an idea, and my frontal lobe wasn't developed, and as a result, I engaged in an action. And I regret it. Now that I'm older, my frontal lobe is developed, and it's working fairly well, and I would never engage in these kind of actions. but at the time I did. And a lot of teenagers engage in stupid actions, stupid decisions. Until you're about your mid-20s, then that kind of excuse goes away, right? And not to say that you can't have, a lot of control and be a teenager at the same time. You can. There's individual differences. And as far as people with antisocial personality disorder or psychopaths or serial killers, there's some evidence that their frontal lobe may be impaired, right? However, we don't do much research on them because A, there's not that many serial killers out, thankfully, out there in the world. And B, once you put them in jail, they're not very they're not very easygoing subjects that you can just walk up to and say, "Hey, you want to be in my experiment?" That's a very difficult thing to do with serial killers. So we don't have much research on them, but there's some thought that their frontal lobe may be not working properly, or people that engage in very hasty actions or very unsound actions without long-term thought may have problems with frontal lobe processing. But brain damage to the frontal lobe leads to this chronic disinhibition of behavior. And that's what we saw in Phineas Gage here. So a lesion is one way we study brain damage in animals. We surgically remove some or burn some brain tissue. So you may cauterize it or burn it out. You may cut it out. But you're removing part of the brain and you're looking to see if there's a change in behavior. And unfortunately, once you remove part of the brain, it's not growing back because neurogenesis is very, very limited. And no one's growing back part of the brain. The brain will rewire itself if there's extensive damage, but it will not grow back. It's not like skin cells where you cut yourself and you'll form a scab and then eventually your skin will heal over the top. Your skin cells will regenerate. Your nerve cells, your neurons do not regenerate. So this is done primarily in animals, mostly rats and mice. No one's using, cats, dogs, monkeys. Monkeys are very close to humans, but they're also very expensive subjects to actually run experiments on, and you don't want to cause damage to monkeys. They're very expensive. So it's performed on animals. It's yielded a lot of insights about the brain, especially less complex structures of the brain. So if you cut out a visual area of the brain, you'll notice a decrease in vision, certain aspects of vision, or temporal lobe, you'll notice a decrease in facial recognition, etc. But very complex brain processes generally use the whole brain and not one particular area. So you may learn something about simplistic processes, but not whole brain associated processes through lesions of individual areas of the brain. This is not as much necessary these days because we have MRI machines that we can study humans without doing lesions on animals. We also have the magnetic cranial stimulation, magnetic cranial stimulation machine, which I mentioned can deactivate parts of the brain when you hold it to the brain, and you get similar information. So what do we use to study the brain in humans? We use 4 tools: EEG, electroencephalogram. These are the electrodes on your skull, on your scalp that measure brain activity. You have MRI, magnetic resonance imaging. It's a 3-dimensional magnetic, image. So you go into a giant magnet. Some of you probably have experience doing this. It sounds like a jackhammer going off constantly, and you go into the MRI machine for about a half hour to 45 minutes, and you get a very good high-resolution 3-dimensional image of soft tissue, like brain, or like the discs in your back, if you have a back injury, or ligaments and tendons in your knees, etc. So soft image, not bone. This is mostly for soft tissue, I should say. Very detailed image of soft tissue. And then you have fMRI, which is functional MRI. And functional MRI is your traditional MRI, but with brain activity on top of it, almost like creating a movie. So you can see the changes in brain activity over time. If you take a picture of the brain, an MRI of the brain, and then you overlay the fMRI signal. The fMRI signal is technically a measure of oxygen use. They call it the BOLD signal, blood oxygen level dependent signal. It's the amount of oxygen each part of the brain is consuming at a given time. And the more oxygen consumed, the more active that part of the brain is. And so you can tell that by the different colors. The brighter the color on the fMRI, the more active that area of the brain is. And the darker the color, the less active. And you can see, you can actually add and subtract the levels of activation to see if there's differences in activity depending on the condition of your experiment. So as I mentioned, EEG is a bunch of electrodes. If you ever had a sleep study done, this is what they do. They put electrodes on your head and they monitor the brain waves. And each brain wave refers to a different stage of sleep, and they can actually see how much sleep you're getting in each particular stage. So it's very effective for studying sleep disorders. It's also effective for seizures, because a seizure will show up as very active and irregular patterns on the EEG. So they can see how long the seizure lasts, what kind of seizure activity you have, and they could possibly localize the seizure. Although it's difficult, it's easier to localize dysfunctional brain activity with MRI or PET scan, but you can localize to a degree off of EEG. But sleep and seizures are the main thing it's useful for. PET scan is useful for studying tumor growth because what it does is it measures glucose consumption. Glucose is the body's fuel. Tumors are highly metabolically active. They consume a lot of energy. Therefore, they consume a lot of glucose. And a radioactive dye is given to you in an IV. And this radioactive dye binds to glucose. And then you could see the radioactive dye showing up as activity on the brain scan, on the PET scan. So the brighter the color, the more radioactive dye is in that location, which means the more glucose that area of the brain is consuming. And you could tell how big the tumor is, how active the tumor is, how fast it's growing in like more aggressive forms of cancer. But PET scans are very helpful for detecting the rate of tumor growth or the rate of cancer growth, especially in the brain. Here's your MRI photo. This is what an MRI looks like. It's a high-resolution three-dimensional image formed by a giant magnet. The MRI machine is a giant magnet. And so if you look at the picture on the left, you see ventricular enlargement. Actually, on the right, I should say. Look at the picture on the right. You see ventricular enlargement. Ventricles in the brain are empty spaces. So the person on the right has large empty spaces in their brain, ventricles. And this is associated with schizophrenia. This is basically brain death. You also see a similar finding in chronic drug users and alcoholics. On the left, you see a normal control. You see how small the ventricles are, those empty spaces next to the arrow? You see how big they are in the schizophrenic patient on the right. So there's actually brain death occurring during schizophrenia, which is really interesting. So fMRI, as I mentioned, looks at blood oxygen levels in the brain and how they change. And the more active that brain is, the brighter the color it will be on the fMRI because it'll be consuming more blood, higher levels of blood oxygen. And we're studying a lot of things with fMRI these days, and I'll have a couple of videos for you to watch in the module for this week to see, the extent of what fMRI is doing for us today. Now we're going to change course and talk about the old brain. So I'm not going to talk about imaging anymore. Now we're talking about the older parts of the brain, the brainstem, the cerebellum, responsible for movement, breathing, etc., very basic functions. The limbic system is responsible for basic emotions, fight or flight, anger and or anxiety. And then later on, we'll talk about the cortex in the next module. It's the outer covering of the brain, that wrinkled mass that makes the brain look kind of weird looking and squishy and all folded and convoluted. That's the cortex. Cortex is what separates human beings from other lower level primates like mice and rats and dogs and cats, etc. So if we look at the brain stem, you also have some other basic brain structures. So in the brainstem, you have the pons and the medulla, very important, vital structures. Then you have the thalamus, which is a sensory relay station. You have the reticular formation, which is necessary for keeping awake and aroused. The cerebellum, responsible for movement, and the limbic system for basic emotions. So starting with the brainstem, as you can see here, at the base of your skull, at the back of your skull, you could probably feel it. You have a little bump. And then, connecting your skull to your neck, you have the brainstem. Obviously, it's within your body, right? So it's behind the vertebrae, so it's protected. It's in your skull, at the base of your skull. And it's there for a good reason, because it's a very sensitive area, and any damage to this area will create long-term problems. Let's start with the thalamus. The thalamus lays on top of the brainstem. The thalamus is the sensory relay station. So all of your senses, with the exception of smell, go through the thalamus. The thalamus is kind of like your junction. It's kind of like Grand Central Station. That, you arrive at Grand Central Station and then you're rerouted to various destinations in New York. The same thing happens in the thalamus. The sensory information first goes to the thalamus, and the thalamus reroutes all that sensory information to different parts of the cortex, with the exception of smell. Smell does not go through the thalamus. It goes directly to the cortex. I don't know why. That's just the way we were designed. Now, if you look below the thalamus, you have the pons. The pons is responsible for coordinated movement. Eye movements especially. Unconscious. You don't control your eye movements to a degree. Your eyes are constantly moving back and forth ever so slightly. They're twitching, and the reason why they're twitching ever so slightly is so that the retinal image does not become stabilized, because if the eye never moved at all, the image would become stabilized on your retina, and you would habituate to it, which means that the image would disappear, and you would no longer see something until you moved your eye again. So the eye has to constantly be moving it very, very slightly so that the retinal image is not stabilized. And the pons are responsible for that. The pons is also a problem in that if you had selective damage to the pons, you would end up with something called locked-in syndrome, which is a horrible, horrible condition where you're alive, your brain is working really well, your eyes are moving, but you are completely paralyzed. The rest of your body is paralyzed. So you can't talk, you can't, move your face in any way, you can't move your body in any way. So you're there, your eyes are open, but you can't communicate, but you're fully conscious of everything that goes around you. And so in locked-in syndrome, they may mistake you for being basically in a persistent vegetative state, or in basically brain dead. And there have been cases where they've actually tried to pull the plug on people that were locked in. But the person that's locked in is still alive. They know what's going on, and they're like, don't pull the plug, don't kill me. But no words come out of their mouth because they can't talk. And as a result, it's a really scary situation. There was actually one patient who wrote a book. I think it's called The Bell and the Butterfly, if I remember correctly. It's something to do with butterfly. But he wrote a book about his experience, and he wrote the book by just moving his eyes. So he moved his eyes to different letters on an eye chart, and then just kind of blinked out the book, because he can't talk and he can't type. He blinked out the book through his eye movements, and to write about his experience, which is absolutely amazing. And they made a movie, I think, about it too. He died shortly after that, though. He was alive for a few years. There have been people that have been locked in that actually recover fully. And we don't know why they recover. We don't know why certain people recover and certain people don't. It's just the way it is. And it's a really scary state, but people can learn to communicate by eye movements. But imagine doing that your whole life. It's just very, very scary. So that's pons. And then you have below that is the medulla. We don't have people with damage to the medulla, because if you have damage to the medulla, you're probably dead. The medulla is responsible for heart rate and respiration. And, that's basically keeping you alive. So if there's damage to the medulla, you're having a severe problem. And the reticular formation is part of the brain that runs through the brainstem. It's a band of fibers that's responsible for keeping you alert and aroused. And if damage is to the reticular formation, you will then basically go into a coma and never wake up. And they've done these experiments in cats where they've cut the reticular formation, the cat goes into a coma and never wakes up. As I mentioned before, medulla is controlling heart rate and breathing, and if damage to the medulla, you're basically dead. This is a big problem. Pons is for eye movements that are unconscious. The tiny eye movements are eye saccades that you make to keep the retinal image unstable, right? And the thalamus is the sensory relay station or switchboard for everything except smell. And the reticular formation is a network that enables us to be alert, aroused, and any damage leads to a coma, as they've done in cat experiments. Adjacent to the brainstem, you see this little ball, or little brain they call it. It's called the cerebellum. The cerebellum is responsible for voluntary movement, like playing a sport or playing an instrument, complex motor movement. The cerebellum is also responsible for procedural memory, or what is called implicit memory, motor-based memory, and classical conditioning, which we'll talk about later in the course. Selective damage to the cerebellum is not very common, because if you had damage to the cerebellum, you probably have damage to the brainstem, you're probably dead. So it's really hard to examine, site-specific damage to the cerebellum because it's right next to the brainstem and right next to these vital structures that we can't live without. In the case of Ronaldo, he can learn to shoot towards the goal because of the cerebellum. He can't learn to pass the ball. Sorry for all you Ronaldo fans. He can't learn to pass the ball, but he can certainly learn to shoot and score the goal. Now, slightly above the brainstem and the cerebellum, but still within the core of the brain, we have the limbic system. The limbic system consists of the hypothalamus, the pituitary, the amygdala, and the hippocampus. Emotions such as fear and aggression, the fight or flight response, is coordinated by the limbic system. You also have basic drives such as hunger and sex, and the formation of episodic memories. The hippocampus, which is that seahorse structure that you see on the slide, it goes all the way around, that processes episodic memories. Episodic memories are memories of events or memories of details. So like what you had for breakfast today, or what you learned in the course yesterday. These are episodic memories. And the hippocampus is responsible for processing episodic memories. The hippocampus can work with the amygdala to form emotionally charged memories. These are called flashbulb memories, when the hippocampus works in tandem with the amygdala. The amygdala is the emotional component of the limbic system. The hippocampus is the memory component. When they work together, they form these really deep, strong emotional memories called flashbulb memories. A flashbulb memory would be like, what was your memory of the day that 9/11 occurred? And maybe some of you are very young. I was a little older. I can remember the exact details. I knew that my cousin worked in the World Trade Center. I had a very, very vivid recall of exactly what was going through my head, and that, we were calling the office and there was no response, and I didn't know if my cousin was dead or alive, and we're calling her house, so there was no response. And fortunately for all of us, she didn't go to work that day, and she was sleeping and was not aware of, this was early in the morning, around 8:30 in the morning, and she wasn't aware of what was going on at all. And unfortunately, she lost a lot of people that she worked with, and I know many people that didn't get out of the building, and some that did, and the some that did are still traumatized to this day. Your parents may remember, or your grandparents may remember the assassination of JFK, exactly when it happened, exactly what was going on, and the sort of malaise that hit the country at the time. So when the hippocampus works with the amygdala, you can form a really supercharged, powerful emotional memory called the flashbulb memory. The amygdala by itself processes emotion, primarily anger and anxiety, or aggression and fear, fight or flight. The hypothalamus is here to regulate body temperature and sex drive, primarily. And the pituitary gland is the master gland of the endocrine system, as I mentioned in the previous module. So once again, looking at the amygdala, if you take a cat and you stimulate one part of the amygdala, the cat becomes very aggressive and ready to attack. If you move that electrode a very tiny bit on the amygdala, that cat cowers in fear of a mouse, becomes afraid of a mouse. It's amazing that you can move the electrode a very short distance on the same brain structure and elicit the complete opposite responses. Aggression versus terror. The hypothalamus, which lies below the thalamus is the sensory relay station that lies on top of the brainstem. It regulates body temperature. So if you take the hypothalamus and you stimulate the outsides, the lateral hypothalamus, an animal will eat and keep eating and never get full and become a giant fat rat. They've actually made obese rats this way that are like 4 or 5 times his normal body weight by stimulating the lateral part of the hypothalamus, the outside part. If you move the electrode a tiny bit and you stimulate the ventral medial part, or the middle part of the hypothalamus towards the bottom, the ventral medial hypothalamus, if you stimulate that area of the brain, the rat will stop eating and never eat and have no appetite and become anorexic. And so the question is, why does one animal become fat and why does one become anorexic, and how can this help pharmaceutical companies? And it's a tricky answer because the hypothalamus also controls body temperature and sex drive. And you don't want to mess with body. You could mess with sex drive. You might upset some people, but you don't want to mess with body temperature because you're going to kill people. And so drugs to just target the hypothalamus have some inherent problems. And so they haven't been developed. So there really is not a good drug treatment for both anorexia or for obesity. The pituitary gland, as I mentioned, controls the whole endocrine system. And the hypothalamus controls the pituitary gland. So indirectly, the hypothalamus then controls the endocrine system. And the hypothalamus can also serve as a reward center in animals. Here's a review slide of the basic brain structures. You should know this slide in depth. You should know amygdala, hippocampus, corpus callosum, thalamus, hypothalamus, pituitary, reticular formation, medulla. Obviously, we all know the spinal cord, cerebellum. And then in the next chapter, I'll be leading off with the cerebral cortex, or the outer covering of the brain. So please review this slide. That takes care of this module, and I'll see you soon. Bye-bye.